Hi everyone, welcome back to our lecture series. Today we'll be looking at nucleic acids, how structure conveys information. So the basic thing we're going to be looking at in this chapter is we'll be looking at the chemistry of nucleic acids, from the nitrogenous or the nucleobases to the components of the general nucleic acid being made up of a nucleobase, a phosphate group, and a ribose sugar. So those are the things we're going to be talking about in this chapter, and then we'll now conclude by looking at the central dogma of biology, and then that will help us to understand our biochemistry even more and more. Let's take it off from there immediately. Okay, so as I have done in every other biomolecule that we study, the first thing we need to understand is what are these nucleic acids, and then what are their functions, if we can understand. Of course, nucleic acids are the basic of information transfer in every living organism. That is the blueprint of our identity. And if you look at my structure here, I got this from Wikipedia, or I should have written that. I got this from Wikipedia. This is from Wikipedia directly. You see the structure of the DNA being made up of two strands, wound on each other, and the RNA being made up of just one single strand. And then each of them are made up of what we call the nucleobases or the nitrogenous bases. These are aromatic compounds that contain nitrogens. So the cytosine, the guanine, the adenine, and uracil are usually made for the RNA, whereas the cytosine, guanine, adenine, thymine, are the nucleobases for DNA. Although some of the RNAs, like the tRNAs, also have thymine as well. So let's just talk about their functions. What are the functions of nucleic acid? So I'm going to look at both the functions of nucleotides and then polynucleotides like nucleic acid, and I combine other functions. The first thing we need to, and what we know, is that they store the genetic information, and those are what we find in the DNA and RNA. So that the chemical basis of heredity, if you're going to transfer a particular trait from a parent to an offspring, those blueprints is contained in the DNA and hence the RNA, and then those are what is transferred into a particular type of protein in translation. Then another important function we need to know is that they store what? Chemical energy. The hydrolysis of high energy phosphate, ATP, is the basis of coupling exothermic or hexagonic reactions to endogonic reactions in metabolism that drives all the reactions we see in intermediary metabolism. So ATP itself is a nucleotide. It is adenosine triphosphate. It is a nucleotide on its own. Number 3, the first structural part of most coenzymes. Coenzymes, like we discussed earlier, are those molecules, those vitamin derivatives that help enzyme to perform its functions. What do? They either carry groups or they transfer electrons from one substrate to another. And that is exactly what we see at this point. So now each of these guys are made up of some part of a nucleotide, like NAD, which is nicotinamide adenine dinucleotide. You see the nucleotide name, FADH, flavinoadenine dinucleotide. Both of them, I wrote them in the reduced form. NADPH, nicotinamide adenine dinucleotide phosphate. And then even coenzyme A all have this nucleotide portion in them. So the important structural forms of these enzymes. So indirectly, you can also say they act as coenzymes. That is very correct. Again, important thing we need to know is that they act as second messengers in signal transduction. You see, most polypeptide and peptide hormones function through a secondary messenger. There are a lot of them, but one of the most common second messengers is what we call the cyclic AMP, or you can say cyclic adenosine monophosphate. Is an important signal transduction molecule in our body. Again, you see how important these things are. So they help this important function make sure that these hormones, these endocrine hormones, carry out their signals and as a result bring about a biological effect. Again, important information about the DNA. The DNA helps direct the synthesis of the RNAs. Remember, we have so many types of RNA, but most importantly in them is the messenger RNA, which is usually carries that coding information. then there is the transfer RNA, and there is the ribosomal RNA. And then finally, they also direct the synthesis of proteins and polypeptides. Remember, the expression of a particular genetic trait is actually resident on the type of protein or polypeptide being synthesized. And that is actually what we see in the central, we're going to talk about this lastly, in the central dogma of molecular biology. This is the central dogma of information transfer. In fact, this is much more related to these hereditary, to the hereditary information here. So whereby RNA is replicated, that RNA now directs for the synthesis of particular RNA, and let me start afresh. I said RNA. A DNA is replicated. Now the replication of DNA directs a synthesis of RNA. It is this RNA that directs a particular synthesis of a protein. Now, a protein can be a hormone or an enzyme that has a particular biological function in the body. So these are the functions of nucleic acid and nucleotides. And now take it out from there. So what is now a nucleotide? A nucleotide simply means a nitrogenous base, or what we call a nucleobase, a sugar, mainly of the ribose or deoxyribose, and a phosphate group combined together. That is what makes a nucleotide. Now, what are the nucleobases? That's the first thing, because we're going to start trying to break down these three components of the nucleotide. A nucleobase are those aromatic compounds that are of two types. We know the nucleobases are nitrogen-containing aromatic compounds that they make up the coding portion of the nucleic acid. There are two types, either the pyrimidine or the purine. The pyrimidine bases are derived from pyrimidine ring. The pyrimidine ring is made up of a 6-membered aromatic ring, and they are the parent compound for the cytosine, the thymine, and the uracil. We're going to see this in the next page. And then all the purines. The purines are compounds that contain a 6-membered ring that is fused to another 5-membered ring. We call them the purines. So they derive from the main parent, which is purine. That's what we call them the purine base. It is the parent of both the adenine and the guanosine bases. So we have 3 pyrimidine bases and 2 purine bases, and that's what we're going to see. Now, let me try to make a demarcation here. Now, to the left are the common bases that we see all the time. These are the most common ones that we see. These are the less common ones, but they exist, and depending on particular point in metabolism. Now, like I said, this is the pyrimidine. The pyrimidine is derived from this aromatic nitrogen-containing ring. It has one nitrogen at number 1, and you count it towards this way, anti-clockwise rather. Now, so we have 3 of them. The 3 of them are the cytosine, the thymine, and the uracil. Now, the cytosine is found in both DNA and RNA. The uracil is only found in RNA. However, the thymine is found predominantly in DNA, but a few of them can be found in RNA. Not many authors actually talk about this, but at least we know that a type of RNA, which is the transfer RNA that plays an important role in translation of proteins or the synthesis of proteins, contain minute quantities of thymine. And then the purine bases, now they contain, you see now, the purine bases contain, this is the part of the pyrimidine part, and they also contain a 5-membered ring, so which are fused together. So this is what we call the purine ring. So the purine ring are just 2 bases. The adenine base, adenine is when you add an amino group at this point, and then the guanosine is when you add a carbonyl point at carbon number 6, and then in number 2 you now move this amino group to carbon number 2. Now, other less common ones are the hypoxanthine, and then N6-dimethyl adenine. Here you have an adenine that has 2 methyl groups attached to it, just like this, 2 methyl groups. That's what, and is in what? Nitrogen is in what? Is in carbon number 6, and this carbon number 6 contains this nitrogen. That's what we call it N6. Here we have 5-methyl cytosine, 5-cis-dihydrouracil. So these are those common distinct, and we're going to be talking about. And then we're going to try to take this to the next level. In the next level, what happens? When you combine, look at now, you see this is separate from this. When you combine a base with a ribose sugar, and it is being connected by a covalent bond. This covalent bond, of course, is a beta-glycosidic linkage. This, what do you form? You now form something we're going to be talking about in the next page. It forms a nucleoside. So a nucleoside simply means a base, a nucleobase, let me see, nucleobase plus what? A nucleobase plus a ribose sugar. It could be ribose, it could be deoxyribose. Remember, the deoxyribose are the ones that are found in what? In DNA. The ribose is in DNA. So when you combine these two together, the form we call a nucleoside. So a nucleoside is a purine or pyrimidine base that is bonded to a sugar. And this bonding is a covalent bond that is linked together by a glycosidic linkage. Usually beta-glycosidic linkage. It lacks the phosphate group. So a very good example here is this one. This is called cytidine. Cytidine is a ribonucleoside, of course, of a pyrimidine. So this is your cytosine connected through beta-glycosidic linkage to a ribose. Now this is these 2-hydroxyribose. Now if you look at this one, this is deoxy because in carbon number 2 of this ribose has been what? The oxygen has been removed. So technically what you did here was to remove this, and that's why you now call it, remove those oxygen, only have hydrogen, you now call it a deoxyribose. That's exactly what you've done at this point. So now you find out that these two guys, look at their name. This is deoxyguanosine, cytidine. So both of them are, this is a ribonucleoside, this is a deoxyribonucleic. So the name of nucleoside, when you add a nucleobase plus a ribose, ends in side for the purines and INE, dine for what? For the pyrimidine. So if you have thymine plus a ribose sugar, you're going to call it what? Thymidine. Right? Uracil will be uridine. If you have you're going to call this one guanosine. If you have this, so this guanosine, so adenine will be adenosine. All right. So if you have adenine plus a base, it's going to be adenosine. If you have thymine, it's going to be thymidine. If you have uracil, we call it uridine. Now we're going to take that to the next level. Now, what is the nucleotide? Now, to extend it, a nucleotide, like I said earlier, will be a nucleobase plus a ribose plus a phosphate group, and that's what we're going to see. So a nucleotide is formed when a phosphoric acid group, a phosphoric acid is a phosphate, has been esterified to the hydroxyl. You're going to esterify to the hydroxyl group of the monosaccharide, most commonly of the what? 3-hydroxy or the 5-hydroxy. And it is named for the parent nucleoside by adding monophosphate. Now see, let me show you this structure. You see, if you look at this structure now, this is 1, 2, 3, 4. So if you connect through, if you take a phosphate group and add at this point, or add it at this point, it's going to become a nucleotide. I'm not going to add it here because this is now so that you don't get confused when you're watching the video. So that's what we call a nucleotide. So if you want to make a nucleotide, remember earlier I said it's called adenosine. Remember, if you have an adenine plus a ribose sugar, we call this adenosine. If you add it plus a phosphate again, phosphate, what is going to be the name? This is what we call a nucleotide. So this is a nucleotide. This is called a nucleotide. And this nucleotide is called adenine monophosphate because the phosphate is 1. So this is going to be adenine monophosphate. That's what you see. So you just add a suffix to it now. Now what then? So polymerization of this single nucleotide into a large big molecule is now what we refer to as a nucleotide. So if an adenine monophosphate polymerizes and keep forming repeating unit of itself, joined together by the phosphodiester bond that is a covalent bond, and forming a long chain, we're going to have a polynucleotide. So a polynucleotide is simply formed from the polymerization of this nucleotide. And the polarity is usually from the 5' end to the 3' end of the phosphodiester bond. Now, this covalent linkage is the covalent, this 5,3 phosphodiester bond is a covalent linkage in which the phosphoric acid is esterified to what? The 3-hydroxy group of one nucleoside and the 5-hydroxy of another nucleoside. And this forms the backbone of all the polynucleotides or the nucleic acid, as we're going to see. So first, let's understand what the nucleotides are. Now, this is guanine. This is the structure of guanine. Remember what I told you? When you add this, it becomes guanosine. If you now add, remember this is carbon number 1, carbon number 2, carbon number 3, carbon number 4. When you now add, okay, this is deoxy because the OH group is not added. Whenever the OH group is not added, this is deoxy sugar. Now, if you see 1, 2, 3, 4, 5. Now, when you add a phosphate group at this carbon number 5, this is the phosphate group, you form, so this now is going to be called guanosine or guanosine monophosphate. That's exactly what this is. And it is a nucleotide. This is a nucleotide, which is the common repeating unit of all the polynucleotides like the DNA and the RNA. So these are some good examples of the nucleotide you're going to see. This is the ones formed from cytidine. It's called cytidine monophosphate, CMP. This is uridine monophosphate, UMP. This is deoxytyrosine. Like I said again, this is deoxy. The OH group is missing. It's just hydrogen. This is deoxytyrosine monophosphate. This is adenosine monophosphate, and this is guanosine monophosphate. So this is also how you name this, naming them from their parent nucleoside and then adding the monophosphate suffix. All right. So now, like I said, polymerization of this nucleic acid, polymerization of this nucleotide. Now, when you take this nucleotide, now look at it. When you take this particular nucleotide, this is the AMP, and now attach it at this point. Attach it from here to this point. It becomes 2. So this now be a dinucleotide. You're not beginning to form a nucleotide. You take another one, add it at number 3. Remember, it started at number 5, and you keep adding at number 3. That's why the polarity goes from 5' to 3' end. That is what we call it. That's the polarity of nucleotide. So let's look at the deoxyribonucleic acid or the DNA. It is a bipolymer of nucleic acid that contains the alternating backbone of the 2-deoxyribose sugar and a phosphate. Of course, the 3-hydroxy group of one of the 2-deoxyribose is joined by the 5-hydroxy of the next one. Remember, in this case, the ribose sugar has been reduced. The oxygen has been removed. Sorry, this is H. Here, this is OH. So this is what makes it deoxy, because the oxygen has been removed. So removal of oxygen, remember, is what? Reduction. So it has been reduced. That's what that simply means for the ribose sugar. Now, the nucleotide residue of nucleic acid are numbered from the 5' end to what? The 3' end. In one system, you can use, usually we try to number them using the abbreviations for the bases. Adenine is A, guanine is G, cytosine is C, uracil is U, and thymine is T. It represents the individual bases. And the vertical line shows the position of the sugar moieties in which the individual bases are actually attached. I'm going to show you that notation as we move on. Now, the diagonal line through the letter P represents the phosphodiester bond. Remember, that bond that is being formed that holds them together is what we call the phosphodiester bond. So common system of notation uses only single letters to show the order. Like I told you, some of them use like the single letters A, G, C, U, T. And then an example would be PA, means 5' end of adenosine monophosphate. That's exactly what this means. Now this differs from RNA. Remember, in RNA is very different, because the sugar the hydroxy group in number 2, the OH in number 2 remains intact. It is a ribose rather than the deoxyribose. So in writing that of the ribose and writing that of the deoxyribose, you can put D to represent deoxy, could be added to the deoxyribonucleic residue. Example, if you say D, this means deoxyguanosine monophosphate. That's what it means. Substituted for G. So if you say this, what it means is that all these are the deoxyribose sugars for the DNA molecules, because all you find in the DNA molecule is the deoxyribose sugar and not the ribose sugar. And let us see this picture. This will make a clearer understanding. Now, this is a polynucleotide because it's more than one. So this is the first one, 1, 2, 3, and 4. So this is a tetra. You can say this is a tetranucleotide. So it's a portion of both. We call this a tetranucleotide portion of the DNA chain. Now, what you see, you find out that now the phosphate group is attached at this end. It's free. That's why we call it the 5' terminus or terminal. That is the terminal. And now this is the first one. Is attached through the. Now there's a hydroxy group here that forms the phosphodiester bond with phosphate here, and this phosphodiester bond is connected to 5' end of cytosine. Remember, this is adenosine monophosphate connected to cytosine monophosphate, and this cytosine monophosphate is also connected to guanosine through this 3' end, and this again is connected to this, forming those there. So this phosphodiester bond, remember again, this bond is what? Is a covalent bond. And in fact, this is the bond that maintains the primary structure of nucleotide. This is the bond that maintains the primary structure of the nucleotide. The primary structure is being maintained. This is like when we looked about protein, this is like that single chain of nucleotide being sewn together. Now, and if you look at this now, this is what you find in the RNA, because these are all ribose sugar. So this is what you find in the RNA, because this sugar is not reduced yet. The oxygen is still intact. When we go to this one, look at this one. You find out here now, it has been reduced to hydrogen. The oxygen has been removed. And this is typically what we find in DNA as well. So this is thymine again. Thymine here is connected to guanine. This thymine monophosphate to guanosine monophosphate to cytosine monophosphate to adenine monophosphate. So in one of those notations, look at what happens here now. Now this is ACGU. This is exactly what we have here. Now it starts, the P here means it starts at the 5' end, and this shows you the covalent bonds from the 3-hydroxy to the 5-hydroxy of the other one until it ends at this point. So if you look at the polarity goes from 5' to the 3' end, just like you see when you run this thing down. If you run this thing down again, if you draw a line, I don't have enough space to draw the line, but assuming you're going to draw a line again, you find out that this is how the polynucleotide chain runs in both the DNA and the RNA. So what is then different levels of structure of the RNA? I already said part of this already. Now, the phosphodiester bond is what maintains the primary structure of the DNA, which shows you that long chain of the nucleotide molecules that runs from the 5' to the 3' end. So the levels, just like we did for protein, there are 4 levels of DNA structure. The first is the primary structure, which is the sequence of the bases along the pentose phosphodiester backbone of the DNA molecule. And then the secondary structure is the other 3-dimensional arrangement of the nucleic acid strand. Remember, DNA is usually made up of 2 strands that are wound or wrapped around each other. So when this DNA wraps around each other, what maintains that shape is the hydrogen bonding between the complementary bases, as we're going to talk about in the next few pages. Now, the tertiary structure is the 3-dimensional arrangement of all the atoms of the nucleic acid. And in this case, we usually refer to this as supercoiling of DNA. This is not just the normal DNA strand winding on each other. This is extra twist being introduced in that winding is what we call the supercoiling. And enzymes that introduce or remove this are actually called topoisomerases, because what they do is to change the topology of the DNA. And the quaternary structure has to do with interaction with nucleic acid with other macromolecules as proteins. Particularly, most of these proteins, we call them the histones. The histones are basic proteins that help that this the DNA wrap around and form the chromatin, which is the basis of heredity, as we're going to see as we move on. So the DNA is a double-stranded helix. We've talked about the primary structure of DNA, which is the phosphodiester bond as it runs through. Now, what about the secondary structure? Now, two polynucleotide chains are wrapped around each other in the secondary structure of the DNA. Now, this fundamental structure this is the fundamental structural motif of the DNA. Now, look at this. One of the polynucleotide chains runs from 5' end and stops at the 3. Look at the green one. And the brown one runs from here 5' end and stops at the 3. Now remember, the bases connect each other. The connection between these two bases are held together by what? Between these two complementary bases facing each other are held together by hydrogen bonds that maintain this three-dimensional structure. Now, because these ones now themselves as a helix, the helix makes two types of grooves. The large groove and what? The small groove, or some people call it the major groove and the minor groove, of course. Now, this particular structure was proposed by James Watson and Franz Crick in 1953. That's what we call it, the Watson and Crick structure of the DNA, and that gave them a Nobel Prize. They discovered this structure of DNA using a modern X-ray crystallography machine. Now, the DNA, now remember again like what I said, the secondary structure of DNA is maintained. The winding of DNA to each other is maintained by the complementary base pairing, and those base pairs are held together by hydrogen bond. That is also another important factor. Remember what I said in chapter 1, we talked about the importance of hydrogen bonding in biomolecules. It helps to maintain their particularly secondary structure, so that the moment those hydrogen bond is broken, those biomolecules lose that their typical native structure. So the major factor stabilizing the helix is the base pairing by hydrogen bonding between adenine and thymine, AT binding, or guanine and cytosine. So we call it the AT and GC. Now the AT base pairing usually have 2 hydrogen bonds, and it is weaker than GC base pairing that have 3 hydrogen bonds. Now, it also consists, like I said earlier, the major groove, which has larger of the 2 empty spaces in an imaginary cylinder that encloses the DNA structure, and the minor groove is the smaller of the empty space in an imaginary cylinder that encloses the DNA. So look at what I said. This is the minor groove. You see, this only contains few tons. This is what? The major groove, which contains many tons. So if you look at the base pairing, that this is the hydrogen bonding base pairing. If you look at the base pairing between AT, it only forms 2 hydrogen bonds. That is why if you have DNA molecules that contain more GC than this, they will usually be stronger, or they will have what we call higher transition temperature or higher melting temperature than those that contain AT. Because you're going to do more work in breaking 3 hydrogen bonds than in breaking 2 hydrogen bonds. All right. Now, DNA supercoiling, which is the tertiary structure. Extra twist can be added in a DNA. In fact, this has been studied particularly in most prokaryotic DNA, which are secular in nature. Most of these like the bacterial DNA is like secular, is a secular DNA in nature. What happens is a type of double-stranded DNA in which the 5' end is connected to the 3' end of the other one to form a circular loop. So, in fact, there's another name we call this DNA. We also call it covalently closed secular DNA. We call it covalently closed secular DNA, CCC DNA. Because of course, remember again, the phosphodiester bond is what? The phosphodiester bond is a covalent bond. Now, it can undergo both positive and negative supercoiling. Negative supercoiling means when it only has fewer number of turns, whereas a positive supercoiling means when it has more number of turns. So, if you want to form this is a relaxed DNA. If you want to form a positive coil, you have to wind it clockwise. If it goes clockwise, it becomes right hand. Look at it. So this is a right-handed or clockwise twist that introduce positive supercoil. And then when you take it to the left, it is called the negative supercoil. And that's exactly what you see here again. So this is the relaxed DNA, the CCC DNA. Remember, it is called, like I said, the covalently closed DNA. So this is the CCC. No, that didn't come out right. DNA, covalently closed circular DNA. Now, if you introduce a positive coil, look at what happens here. A positive coil, when it winds on itself round, of course, what does this are enzymes. It forms a so this is a positive supercoil, and this is a negative supercoil, of course. All right. So topoisomerases are enzymes that introduce or relax this supercoiling in circular DNA. And there are two types. There is the class 1 and the class 2. The class 1 cut the phosphodiester bond on one strand and reseal it again. That's what they do. It cuts it from one strand and reseals it up. In class 2, cuts the two strands, reseals them to introduce the coil. And a very good example of one of these DNAs is what we call, commonest one that have been several studies, is the DNA gyrase. It is a bacterial topoisomerase that introduces negative coils in DNA during replication. What it actually does by introduction of negative coil is to relax the positive coil and let it have fewer number of coils, and then from there transcription can take place. So this is the DNA loops, and what the gyrase does is that it binds this, cuts one of these, and then reseals it and introduce, look at what other point in here, it introduce more supercoils, because that is what this enzyme do, is to introduce supercoils into a relaxed DNA. All right. So supercoiling in eukaryotic DNA now results to even a larger quaternary structure. In this place, it results in what is called a chromatin. A chromatin is a complex of DNA and proteins. Most of these proteins, we call them the histone in eukaryotic nuclei. Here, topological changes are induced by supercoiling that must be accommodated by a protein. We call them the histone protein. The histone proteins are basic proteins that are found complexed or in association with the DNA. They're complex with the DNA. And they are rich in lysine and arginine, which are basic amino acids. They are highly basic proteins. So, and there are many types. There is a H1 histone, H2A, H2B, H3, and H4 histone. So, they help in wrapping DNA around these proteins. And when they wrap around these proteins that looks like a cylinder, they form a complex which is chromatin. And this is what you're going to see. Now, the basic structure of the chromatin resembles a bead on a string. You know, when you have your necklace as a bead on a, oh no, that is not what I want there. I'm going to remove it. When you see a bead, you connect a bead in a string, and you wear them as necklace. That is what this looks like. Now, so we found out each of this is the DNA that is wrapped around what? The histone protein. So this is the histone protein wrapped around it. So it resembles a bead on a string. Each chromatin is a nucleosome. Each bead becomes what we call a nucleosome. It's a globular structure in which the DNA is wrapped around an aggregate of histones. Remember, the histones are this protein. So this is the core of 8 histones. This is the histone on the, I think this should be purple color. I'm not very good in color. This is the histone, and then the DNA is in yellow here, wrapped around it. So that simple wrapping around of a protein and a histone is what we call what? A nucleosome. It's a globular structure in which the DNA is wrapped around an aggregate of a histone molecule. The strings portion of without the histone is what we call, they are called spacer regions. And recent research has shown that the structure of the spacing of the nucleosome is important in the function of chromatin. So this is how the DNA is wrapped around and then placed inside the nucleus. All right, so this is a single histone molecule being held together by the what? A single histone molecule holds the DNA core at this point, like a bead. So this is the structure of the chromatin. Now, denaturation of DNA, just like any biomolecule. Any biomolecule want to exist at a what? At an optimum pH, an optimum temperature, above or below which is going to be destroyed, and the native structure will be destroyed and is not going to perform its biological function again. So energy must be added to a sample of DNA to break the hydrogen bonds and disrupt the stacking, the base stacking, of course, that are held together by hydrogen bond. And this can be carried out by either heating the DNA in a solution. Heat denaturation of DNA is called melting. It's actually called melting as it is, because that's exactly what it means. Now, of course, the bases will absorb light at 260 nanometer wavelength. So as the DNA is heated, the strands will separate, and the wavelength of absorption actually does not change, but the amount of light absorbs increases, and that leads to the separation or the breaking of those complementary hydrogen bonds, which are between AT or GC. Remember, this only has 2 and this has 3. Now, what is the implication of this? The implication is that the melting temperature, midpoint of melting curve, will be higher in DNA that contains the GC than those that contain the AT, because in GC you have to break 3 hydrogen bonds. Renaturation is also possible like in proteins. In this case, when you begin to cool them slowly, the DNA can form. So this is the native double helix. When you begin to heat it, it will begin to break the hydrogen bonds at this point, and they will now begin to separate. If you add more heat, you can now separate it into the 2 strands of different DNA nucleotide. And then if you begin to cool it slowly again, it can renaturate or begin to reform. The hydrogen bond can reform once again and assume the native state. Now, the RNA is different from the DNA because the RNA is just made up of only a single strand. It consists of long unbranched chain of nucleotides joined together, the same again, from 3 hydroxy of one pentose to the 5 hydroxy of the next. The pentose unit here is not the 2 deoxyribose. It is just the ribose itself. In these 2, there is the OH group. The OH group is there. In DNA, there is the O has been removed to form the 2 deoxyribose. Again, the pyrimidine base that are predominant here is the uracil and the cytosine. The thymine only exists in one type of RNA, which is what we call the transfer RNA. Now, of course, this is what we call the fundamental process of information transfer, the central dogma. Now, the DNA replicates to give birth to itself. That's what we call DNA replication. A DNA can replicate to produce other forms of DNA. This is DNA replication. Now, DNA cannot transcribe itself again to produce the messenger RNA. We call that transcription of DNA to RNA. And then different messenger RNA can now be translated to produce the polypeptides or proteins. Long polypeptide proteins can now be produced from the information, of course, they get from this DNA. And this process is for converting from mRNA to proteins is what we call what? The translation. So this 3 pathway is what we call the central dogma of molecular biology, of information transfer. And this is why we call nucleic acid the informational blueprint of every organism. Now, what are the roles of there are different types of RNA we're going to talk about. Now, we're going to talk about the most important ones and other ones that are not usually mentioned. So, here are the roles of different kinds of RNA. We're going to look at the size and we're going to look at the function. The transfer RNA, it is small. What does it do? It transports amino acids at the site of protein synthesis. At that site of protein elongation, in translation, it is the transfer RNA that brings every amino acid and synthesizes the peptide bonds. The ribosome RNA are different kinds. It has different sizes. Now, what it does is that it combines with proteins to form ribosomes, and it is at this place that protein is synthesized. It's also another important major one. The messenger RNA is variable in size. Again, it is this guy that directs the synthesis of protein, because it is it that carries the blueprint or the information from DNA and gives that information to for the synthesis from amino acid to a particular type of protein. The small nuclear RNA processes initial messenger RNAs to its mature form in the eukaryote. You only find this in the eukaryote. Then the small interfering RNA, this one's also not commonly talked about. It affects gene expression and is used by scientists to knock out genes in particular studies. So particularly from here down, are not usually mentioned, but I want you to know the functions of these four, very, very important for enzymes. All right, so these other ones are not too important, are not too mentioned, but if you're going to talk about the information transfer, you have to mention particularly the transfer RNA, the ribosomal RNA, and the messenger RNA. Now, the transfer RNA, let's look at it a little bit, because this place, if you look at my syllabus has this is the transfer RNA. It runs from the 5' end and stops at what? You see that? The 3' end. It is single-stranded polynucleotide chain of 73 and 94 nucleotide residues long. It carries an amino acid. Remember I said it is what carries the amino acid and delivers it to already existing polypeptide and then increases the chain length one at a time. Carries an amino acid at each 3' site end, and then it has intra-chain hydrogen bonding. This yellow light represents intra-chain hydrogen bonding that maintains its native structure. All right. So the next thing there is the ribosomal RNA, or the RNA. That's the shortcut, the RNA. Now, the ribonucleic acid found in ribosomes, it is the site for protein synthesis. Of course, only a few types of RNA exist in the cells. It accounts for about 60 to 65% of the total weight of the ribosome. In fact, the ribosome is just made up of particularly the RNA, the ribosomal RNA. And then the protein portion is about 35 to 40% of the weight. In both prokaryote and eukaryote, the ribosomes consist of two subunits, one large one and small large one. So analytical centrifugation is used to monitor the dissociation and the association of these ribosomes. Of course, you can use that to separate them and use them for studies. Now, the messenger RNA. Now, this is initially formed as a larger precursor of the molecule from the heterogeneous nuclear RNA. Now, it is the ribonucleic acid that carries the genetic information from the DNA. This is the first one that is synthesized from DNA, of course, to the ribosomes for the synthesis of protein. It is present in cells relatively in small amount, and it is very short-lived. The biosynthesis is directed by the information that is encoded by what? The DNA. So the complementary strand of the messenger RNA is synthesized along one strand of an unwound DNA, starting from the 3' end. And actually that is the function of the messenger RNA. It's one of those ones you have to know the important function. The small nuclear RNA is just recently discovered. Still a little bit of subject of debate as well. It is found in the nucleus of eukaryotic cells. It contains about 100 to 200 nucleotides. And then it complexes with proteins and forms small ribonucleoprotein particles. Now, small ribonucleoprotein particles are the protein RNA complexes that are found in nucleus. And what do? They help in processing RNA molecules for export into cytoplasm. They also help with processing of the initial messenger RNA that is what has been transcribed from DNA into a mature ones. Because if initial transcription, whereby the DNA sends information, what the DNA does is to first of all give its blueprint to the formation of this messenger RNA. It is from this messenger RNA now that the proteins are formed. And for these proteins to form, the tRNA will now have to deliver amino acid to already existing chain and now begin to form these proteins. That is how these proteins are actually synthesized. All right. And with this function of small nuclear RNA, we've come to the end of this lecture. Thank you once again for listening, and do have a wonderful day ahead of you. And bye at this point.